Good day, this is Professor Will, CBMD PhD. And today's Wednesday, March 30th, 2022. It's 2:17 and 18 seconds, AM Eastern Standard Time. Today's research, the Fallopian tube. So let's hop to it here. The surprising diversity of the Fallopian tube. A study provides a new cell atlas of the female reproductive organ. Date March 28, 2022. Source, Michigan Medicine, University of Michigan. A new cell atlas paving the way for new research into infertility and other diseases affecting this organ, including some cancers. Using tissue samples from 4 premenopausal women from the Department of Human Genetics, analyzed almost 60,000 cells by single-cell RNA sequencing. They used the data to characterize the diversity of cells that make up the fallopian tube, including both the lining of the tube, the epithelium, and the deeper stromal layer, consisting of immune, blood, muscle, and other cells. It was known that there are about 4 epithelial cell types in the fallopian tube. Researchers were able to reveal a deeper level of heterogeneity within these cells. And specifically, they identified 10 epithelial cell subtypes, including 4 of the finger-like ciliated cells, responsible for moving the egg to the fallopian tube's 3 sections before and after fertilization. The cells within the fallopian tube are ever-changing, replenishing themselves over time and varying in number depending on a woman's age, hormones, menstrual cycle, and in the presence of disease. By comparing cells from women with healthy fallopian tubes to 2 samples from women with a fallopian tube disease known as hydrosalpinx, conventionally known as a blocked fallopian tube, the researchers were able to pinpoint which cells increased in number and which changed characteristics such as high degree of inflammation. And some of the cells are the cause of the disease state, and some others are the consequence. And now we know the patterns for individual cell types to figure out the molecular reasons for that pathology. The team also found that some of the cell subtypes they define in the fallopian tube may function as precursor cells, those that can regenerate multiple cell types in response to normal tissue turnover or for repairing a damage. One of the most surprising findings of the study was the discovery of cells with markers for epithelial mesenchymal transition, a process not previously associated with the fallopian tube, through which a cell can under certain circumstances become cancerous. Ovarian cancer, it turns out, may be a misnomer. The new study adds to accumulating evidence as a root of ovarian cancer. The 5th leading cause of cancer, death in women, may originate within the adjacent fallopian tube. The epithelial mesenchymal transition process seems to be tightly regulated in the premenopausal women. One possible connection to cancer is that when there is misregulation in this population of cells, in some unfortunate individuals, they may develop ovarian cancer. With epithelial mesenchymal transition cells in the fallopian tube, you do have a predisposition right now. Additional insights came from the fallopian tube cells from women with hydrosalpinx, specifically that the disease may lead to a type of scarring called fibrosis. The implication is that for women who don't want to have their tubes removed, you can think about treating them with anti-fibrotic drugs, and such as ones used to treat lung fibrosis as a way to save their tubes during reproductive age. Compared to past efforts, the study provides much more detailed information about cell types and functions in the tube for researchers interested in a host of questions about the normal female reproductive system. And this really is a boot camp to launch future studies, including ones that look at the effects of age, the menstrual cycle, hormone therapy, ancestral background on cellular diversity and disease pathology. Good day, this is Professor Will, CBMD PhD. on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have an awesome day.